Hello everyone, my name is Wilson Cristanto and I would like to present you my presentation of my term project about Neisseria gonorrhea. Let's just begin. Here you go. I would begin with the three questions. The first one would be, how do I know if I get gonorrhea? The second will be, how does gonorrhea work? The third one will be, how to counter gonorrhea. Let's just begin with the first one. So how do I know if I get Neisseria gonorrhea? The answer will be by the signs and the symptoms. So two things. In 1 to 14 days, the person who got gonorrhea will have a signs and symptoms of white, yellow, green discharge, and also burning sensation during, burning sensation and a clap sensation during urinating, which is bad. And for women, it has a dysuria, increase more on vaginal discharge, and also more on vaginal bleeding during period for women because of the infections. If the infections becomes more radical, it appears abdominal pain and can cause fever because radical, it's spreads everywhere. So what are the complications or another signs or symptoms? Yeah, what are the complications? The complications for women, it can be very bad for women because when it spread to uterus or fallopian tubes, it can cause pelvic inflammatory diseases and also infertility, and which is ectopic pregnancy. It's highly possible. For the men, if it reaches to your epididymis, it can damage the tissues on the epididymis, which can lead to infertility as well. So does that mean that if I got the yellow urine or green urine, I got gonorrhea? No, not so fast, my friend. That can be another STDs that can cause it. So how do I know if I got Neisseria gonorrhea? You can run, the answer will be you can run a test. So how do you run a test? That's gonna be the answer on my next slide. It begins with the agar medium. For STDs, they have a special agar medium. And for gonorrhea as well, they have a special agar medium. If you are rich, have a lot of resource, it has a specific medium, which is the Thayer-Martin medium. It has some percentage that can tell that this bacteria suscept the antimicrobial. But before, let me explain more about Thayer-Martin agar. Thayer-Martin agar has a 3 to 7% carbon dioxide. It has the nutrients and also antibiotics like vancomycin, colistin, nystatin, and trimethoprim. If you open and on the links that I share you on the presentation, you will see there's a bunch of lists of antibiotic with the percentage and which percentage that substep, it can determine the STDs. It has a list for Neisseria, but I don't want to share it because it takes a long, but it's pretty fascinating too on the list. And let's say that I already culture it. I put it on the agar. I get the medium. What you gonna do next? You're gonna need to check it on the microscope. So figure out the cell morphology. So begin with the gram staining, put it the iodine. Just do the experiment of the gram staining. And if you find the result like this picture, two bacteria sticking together like diplococci, and it has a little bit, I would say a violet red, or because of the safranin, or a gram negative, you might tell that this is the unique of the Neisseria gonorrhea. You might tell this is the gonorrhea because, sorry, let me rephrase it. Neisseria gonorrhea, their unique is that the cells are sticking together. That's part of the morphology. Pardon my English. All right, next for the culture will be, for the test will be the acid production. According to the CDC, it has a biochemical test. It has some brand name, and you know what? It's a complicated brand name. I don't want to put it on. But the point is, when you run the acid production, you will see that Neisseria gonorrhea only use glucose. They don't use maltose, lactose, and sucrose. And here's the taxonomy, real quick. The kingdom is bacteria, the phylum is Proteobacteria, the class is Beta Proteobacteria, the order is Neisseriales, the family is Neisseriaceae, the genus is Neisseria, and the species is Neisseria gonorrhea. Just a quick short of taxonomy. All right, the next will be the enzyme substrate test. So upon the test of enzyme, it will appears that the Neisseria will produce hydroxyprolylaminopeptidase. And on the nitrate reduction test, it shows negative. All right, the next will be polysaccharide test. Neisseria gonorrhea did not produce, it will show negative because it didn't produce polysaccharide. And also for DNA acid test too. All right, these are the picture. You can see it. Diplococci. Yep, that's the gonorrhea. Yeah. So how does it work? So how can damage our tissues? Yeah, how does it work? And it begins with the transmission. So when you got the gonorrhea, it will stick to your mucous membrane. And when it sticks to your mucous membrane like this, they will create a colony. And after that, they're gonna invade it inside, if possible. The process of invading, they call it transcytosis because the epithelium phagocyte them. So when it phagocyte, it triggers the inflammation or the immune system responses by macrophage and neutrophils. When it comes, the neutrophil will swallow or phagocyte the bacteria. The beauty of this bacteria is that the bacteria can able to survive, which I will explain more on virulence of Neisseria gonorrhea. So how does it work? How can it become virulent? So it begins with the adherence. It means when that gonorrhea sticks on the surface, it's hard to detect, and then they create a lot of microcolonies which can able to spread to other epithelial tissues. That's one. The second one will be the transcytosis, the epithelial invasion, which is still on the research, but it can be damaged to other tissues, not just only the epithelial surface. And the third one will be when the neutrophils invade trying to kill the gonorrhea, they create a leakage, which become a nutrients for the leakage become a nutrients for gonorrhea due to zinc and manganese. And another problem would be the gonorrhea won't eliminated by neutrophils because because of the gonorrhea has some certain features that can against the neutrophils. So what are the features? The features of the Neisseria gonorrhea, it has its form of pili, lipo-oligosaccharides, porine, protective enzymes, OPA proteins, and also the resistance. But the one thing that I would like to add, that I would like to focus in lipo-oligosaccharide, which is endotoxin that can mask the immune cell recognition. The lipo-oligosaccharide, it's not a biofilm. Lipo-oligosaccharides can also help or support the TNF or tumor necrosis factor, which can damage the epithelial tissues more. And also for the protective enzymes. So neutrophils are famous to produce hydroperoxide, which can damage the bacteria and can also lyse the bacteria whenever they produce a lot of hydroperoxide or probably any other chemical products. But because of the protective enzyme like catalase and MSIRB, it it protects gonorrhea. So yeah, the next will be I still have quite time. So I would like to focus more on the virulence. Another one would be the OPA proteins. So the OPA proteins are a switch. It's like a jacket. So when the culprit kills someone, they change the clothes. That's the OPA proteins. You change it. And the pili, which is the little finger thing, they can change the cell structures which can avoid the immune system. It makes it harder to detect. And for the porine is the nutrient channel for bacteria to get the nutrient, and also can suppress the neutrophil from the oxidative burst and apoptosis, that's how the Neisseria gonorrhea works. And quite a bit dangerous. Next would be how to counter Neisseria gonorrhea. Simple, use antibiotics. The CDC recommends 250 milligrams of intramuscular ceftriaxone and 1 gram of oral azithromycin. This won't repair the permanent damage of the disease, but it just kills the bacteria. I think that's it. Thank you for watching, and I hope you have great presentations. Yay!